Hello, my name is Rishabh Jha and I am a MSc Computer Science student currently working on AI in medicine. Now, in this presentation, we are going to talk about linear programming in medicine, and my topic will be flux space analysis of cerebral energy metabolism in Alzheimer's disease. Now, this presentation is not going to be about machine learning. Instead, we'll talk about linear program in standard form and how it is solved by simplex method in case of biological setting. Now, one thing about Alzheimer's is that it stops before it forgets. What it means is if you take a PET scan measure, it measures how much glucose the brain takes up. In Alzheimer's, that uptake falls years, sometimes decades before the first symptom. So the fuel supply degrades before the memory does. A brain is 2% of the body mass and it consumes nearly 20% of the resting energy. So it has almost no slack. A small cut to the fuel budget propagates. Now, this reduced glucose uptake is a tightening of the upper bound U on one variable, the glucose exchange flux. That is a right hand side perturbation. LP sensitivity analysis says the change in optimal ATP yield is the shadow price of the bound, valid over a range where the basis stays optimal. Here, ATP is the energy unit used biologically. So, this linear programming structure is not just a metaphor. It is a parametric LP question that says how does Z star moves as U falls. Now let's look at that. So here we have a standard form LP where we will try to maximize C transpose X subject to AX less than equal to B, and obviously X is greater than equals to 0. Here X is the decision, C is the price on each decision, A and B are the constraints. The feasible region is obviously a polyhedron, and if the optimum is finite, it sits at the vertex. That is why simplex is finite. It searches bases and it's not a continuing cyclic process. So why medicine? Why the simplex method can or a linear programming can solve this issue in medicine? So there are three reasons for that. The first one is network. So metabolism is in the form of network, which gives the node arc incidence that the structural LP can really solve much better. Similarly, resolving. Biology never solves one LP. It solves thousands of near identical ones. And cost per resolve is what matters. And also the decision factor. Like we ask a yes no question or a binary entry with the LP as the relaxation at every node. Now let's get into a little bit of chemistry from biology and let's join chemistry to a matrix. So as we can see, every reaction is a column, every metabolite is a row. Entries record what is consumed negative and produced are positive. This is the stoichiometric matrix S. Now let's point at the glucose row. R1 is -1, so it consumes glucose. R5 is +1, so it supplies or produces glucose from the outside. Let's look at the ATP row. R1 makes 2 and R2 spends 1. This is a chemist bookkeeping. Now let's see how a LP solver will read it. So like V is the flux vector, component Vj is the rate reaction J runs. Now we'll also see the constant here. The internal pools are not accumulating, so whatever is produced, that is consumed. So S times V is always 0. Now let's see this flux-based analysis as a linear program. So it is maximize C transpose V. C selects the reaction we care about. For example, for microbes, it selects biomass, so we choose maximization of their growth. Similarly, for brain model, it selects ATP production. So changing C changes the biological question, not the algorithm. And it is subject to SV equals to 0. So 1 equality per metabolite, so M equality rows. And here L is less than equal to V, which is less than equal to U. So the two physical facts is in one line. Now, also the linear objective and linear equalities is something that we need to know. So a bounded variable LP, the basis stays M by M, however many reactions there may be. So three guarantees follows, and they are why the field standardized on this. Any local optimum is global. Simplex returns exact vertex optimum, and strong duality gives a certificate. So now let's see how we are going to solve the model. So we'll get back into the reaction matrix from the previous slides. So there are three things beyond S, like C picks R2, the ATP consuming reaction. Every reaction runs one way, so L equals to 0 throughout, and glucose supply is capped U on R5 as 10. So we know SV equals to 0 does what it does by itself. So glucose row forces R1 equals to R5, ATP row gives R2 equals to twice of R1, and this is the case that optimum is immediate, right? Also, the bottom number, because this is the movement, 20 ATP from 10 glucose is 2 per glucose, which is exactly the kind of glycolysis that happens in the cellular reactions. Now, this is a very small illustrative network, not a patient model. So it is a small illustrative way of representing a cellular level of reaction, and it can be reproduced on the entire brain level to understand the entire chemistry using the optimization principles of simplex method. So now let's try to analyze Alzheimer's uh, through the concepts of duality. So previously I claimed at the start that Alzheimer's enters as a right hand perturbation. So where's the proof? So let's see. So we know R5 sits pinned at its upper bound. Its reduced cost is C minus Y transpose S, and it evaluates to exactly 2. For a variable at its upper bound in a maximization, that positive reduced cost is the shadow price of the bound. So one more unit of glucose buys 2 more ATP. Now let's do the sweep. Cut uptake 30% from 10 to 7, let's say, and roughly the scale seen on a PET scan. Now ATP yield falls from 20 to 14, also 30%. Now these two identical percentage, this is the moment like where we see the pass through is linear because the basis never changes across the sweeps. One dual member predicted every point on that line, and L resolved nothing to know it. Now in this toy, the shadow price holds for every U. In a real model, the range is finite. Push far enough, another constraint binds. The basis changes, and then you need a new dual. And again, this is a toy sewing mechanism, not a prediction about the patient in a machine learning way. So now let's see the flux variability analysis. So the real question lies on rarely lies on one flux. It's about the range each reaction can take. And FVA, as I previously did in the assignment using the microbiology concept of E. coli's uh, cell division. So Even in that case, the optimization through FVA, flux variability analysis, was 8.6 times better than the cold interior point method, 31 times faster than the cold first order method, and the simplex steps per warm resolve was 0.05. So the flux variability analysis solves two LPs per reaction, that is the minimum and maximum, that differs only in the objective. And only in the objective vector changes what is the classical warm start case. So now let's talk about the application part rather than the implementation part. So the applicative question is, how does patient data gets into a model whose constraints are fixed by the chemistry? So here S is the same for every human. A generic model says what a brain can do, not what this brain can do. Similarly, we have two fundamental programs or two fundamental models, Gimme and IMAT. So Gimme stays a linear program. It requires a metabolic function to stay near optimal, then minimize a weight sum of absolute fluxes through reactions whose genes are quiet. Similarly, IMAT, it sorts reactions into high and low expression of expression, and it tries to maximize how many agree with their how many reactions agree with their expression state. So again, this agreement is yes no. So we'll have binaries which will be used at every node again by an LP. So now let's see what the LP finds in the Alzheimer's brain through all these implementation. So for that, we have to go to one of the finest research in this field by Stempler and his team in 2014. So they took 363 cortical samples from Alzheimer's patients, and they integrated the expression data into a genome scale human metabolic system. And they compared it through FVA exchange intervals. They controlled it against the Alzheimer's cases and predicted reduced secretion of the biomarkers. And similarly, a systematic knockout search rank perturbation was also done for understanding the relation of drug targets with the kind of energy metabolism in the brain. So this study not only helps us to identify how the brain metabolism mechanism happens, but this also helps us to link between the biomarkers, the various metabolism factors, and the chemical processes inside the brain. And this has whatever we did was a toy example, but it has a higher level implications when this optimization comes up with machine learning and clinical setting. Now, there are four things to take away from here. The very first one is the energy routing failure in case of Alzheimer's, and how this is a network flow problem where we get a simplex method to rescue the problem. Similarly, the flux based analysis, the flow problem as a pure LP, also the LP toolkit as a medical toolkit. For example, we have simplex warm started FVA, MILP knockouts, and duality, how everything can come up together for a model development, for example, an optimization model for Alzheimer's, and also the brain energy metabolism. And the last takeaway here is a faster LP means a faster path from model to therapy. So how can we utilize all of these knowledge in actual clinical setting is also one of the most important factor that we can take from here. And in this project, we have used these references to prepare the content that we have. And thank you so much for listening. And I will look forward to the reviews that I'm getting from my fellow friends. Thank you so much.